This is the second of a two-part or a three-part series introducing you to the functions of the adaptive immune response. In this second part, we're going to focus on how T cells and B cells recognize antigens and get activated. The lymphatic system is the system that connects antigens in tissues to where the B cells and T cells are in the draining lymph node or in the spleen. Lymph nodes are connected together by these lymphatic vesicles noted in this cartoon in green. The lymphatic vesicles drain interstitial fluids from tissues, and once those interstitial fluids enter the lymphatic vesicles, we call them lymph. These lymphatic vesicles then move the lymph towards the lymph node via smooth muscle and skeletal muscle contraction. Important to note, there is no pump that drives this. This is a passive movement that's mediated by skeletal contraction mostly. T cells and B cells are activated by foreign antigens brought into the lymph nodes by lymph, and then the lymph will travel down these chains of lymph nodes until it can re-enter circulation via the thoracic duct or the right lymphatic duct. Now, this is a one-way movement. Plasma enters into interstitial fluid, which drains into the lymphatic vesicles and becomes lymph, which then drains back into the blood and becomes plasma. So when we say plasma, interstitial fluid, and lymph, we're really referring to the same fluid. We're just calling it different things based on where it is. Now, there are a number of lymphocyte structures within lymph nodes, but I only want you to focus on a few of these. So lymph nodes drain tissue antigens. The lymph comes into the lymph node via this afferent lymphatic vesicle and leaves the lymph node via this efferent lymphatic vesicle. And within the lymph node, the most important B cell structures that I want you to talk about are the primary B cell follicle, here in purple, and the germinal center, which is comprised primarily of activated B cells that are expanding rapidly, shown here in the gray. The other zone that I want you to know a little bit about is the T cell zone, or the paracortex in the lymph node. This is the site of T cell activation, surrounds the B cell zones, and is rich in dendritic cells. By and large, I will call these B cell zones and T cell zones, but these are important terms to learn and to remember. And the germinal center is going to be a very important structure for understanding how B cells get activated. This is a picture of a lymph node where we can see both a primary B cell follicle right here, as well as a germinal center that is basically blown up in the middle of this primary B cell follicle. And then surrounding this B cell follicle in this germinal center is the paracortex. And so you can think of these as balls of B cells that are the B cell zones surrounded by the paracortex or the T cell zone. Now, there are some analogous lymphocyte structures in the spleen. The spleen drains blood antigens, whereas the lymph node tends to drain tissue antigens. The spleen is composed of red pulp that contains red blood cells and sinusoids. I'm not going to talk much about this aspect of spleen anatomy, but it also contains white pulp, which includes B cell zones and T cell zones and marginal zones. And the B cell zones are very similar in the spleen as they are in the lymph node. They are composed of primary follicles with naive B cells and germinal centers with activated dividing B cells. The T cell zone in the spleen is called the periarterial lymphoid sheath, or PALS. And this is a picture of the PALS here. So first in the cartoon, you can see this kind of purple area surrounding the arterial is where the T cells are congregated. And then if you look at this as a cross section, you can see that there's this kind of sheath of T cells surrounding this arterial coming through the spleen. Okay, and so that's really all I want you to know about B cells and T cell zones and spleen and lymph node anatomy, that these cells differentiate or distinguish themselves based on what part of the spleen or lymph node they migrate to. Now, I'm going back to our old picture of some bacteria that get into the skin and they activate complement, they activate macrophages, but let's focus in again on dendritic cells. Dendritic cells are constantly phagocytosing these bacteria or other pathogens that may enter into this site, and when they do so, they can become activated. And so here's a dendritic cell that's either infected with a virus or has phagocytosed some of that virus, or maybe it's a bacterial infection that's phagocytosed that bacteria. And it can migrate into the lymphatic vesicle and travel down the lymphatic vesicle to the draining lymph node. Alternatively, viruses and bacteria or their products can also migrate via the lymphatic vesicle in the lymph towards the draining lymph node. Dendritic cells will then migrate into the T cell zones of the draining lymph nodes and activate naive T cells. And this T cell dendritic cell interaction can last for quite a long time. So once a dendritic cell finds a T cell that can recognize the antigen that it is presenting to that T cell, it can form a coupling with that T cell that can last for hours or even a couple of days. And once that happens, this T cell becomes activated and starts to enter a state of rapid cell division and activation that is termed clonal expansion. And as these T cells divide rapidly and expand, they form effector functions. For example, we mentioned helper T cells and cytotoxic T lymphocytes. Another way to look at this is that the frequency of naive lymphocytes specific for any given antigen is quite low, usually about 1 in a million. But the process of activation within the lymph node allows those lymphocytes that are specific for any given pathogen to get activated and undergo this period of clonal expansion so that we can increase rapidly the frequency of lymphocytes that are specific for that foreign pathogen. After clonal expansion, the frequency of activated lymphocytes specific for that antigen may go from 1 in a million to as high as 1 in 50 or 1 in 20 or 1 in 10. And that's sometimes greater than 20 cell divisions within the course of just about a week. This is about as fast as mammalian cells can divide. So it's a very robust process of clonal expansion. Now, T cells, to enter this clonal expansion phase, have to receive activation signals from antigen-presenting cells. And so what I've done is I've drawn a picture of an antigen-presenting cell on top that has an MHC molecule bound to a peptide derived from a pathogen that is interacting with this T cell receptor being expressed by a T cell. This T cell receptor expresses a co-receptor. This is going to be either CD4 or CD8. In this case, this is an MHC class II molecule, and this is a CD4 co-receptor that's helping stabilize this interaction. And then there's all these molecules that as a complex are referred to as CD3. And it is these molecules that mediate the intracellular signal that activates the T cell. The reason I bring up CD3 at this juncture is that CD3 is the defining marker for a T cell. And so when you see a diagnostic assay that indicates CD3 positive cells, what that assay is referring to are T cells. T cells also have to receive other signals. So this is just a summary of what I just said. The TCR interacts with peptide-presenting MHC, the co-receptor stabilizes the interaction, and CD3 transmits the signal into the cell. T cells also have to receive other activation signals, and collectively we refer to these activation signals as signal 2 or co-stimulation. Co-stimulation is required for T cell activation. Antigen-presenting cells express co-stimulatory molecules under conditions of inflammation only. And so these co-stimulatory molecules are signals to the T cell that there's an inflammatory response going on, and it is appropriate, therefore, to make an immune response or become activated in response to the antigen that T cell is seeing. So you can think of it as another layer that is ensuring that T cells are only making responses to foreign antigens and not to self-antigens under conditions of non-inflammation. And these co-stimulatory signals enhance T cell activation and survival. Now, I've written an example of some co-stimulatory signals up here, some of the specific molecules that are co-stimulatory molecules. You will learn more about these later in the year. You don't need to know about these right now. So we've discussed how T cells get activated in the T cell zone. Many of these T cells develop effector functions, and these T cells that develop effector functions can migrate out of the lymph node into inflamed tissues and there mediate their effects. But some T cells will migrate to the B cell zones and enhance B cell activation, and we'll talk about that in the next tutorial slide. So this is just going to be a quick introduction to the next tutorial, which will help us understand how T cells and B cells actually develop functions that eliminate pathogens. Okay, that is it for this tutorial.